Okay, so in this lecture, we're gonna cover chapter 2 from your OpenStax book, Chemical Level of Organization. And this is pretty long PowerPoint, so I'll try to do it like with the, I try to spend no more than 1 hour to do it because I don't want this lecture to be too long. Okay, so what is matter? Matter is anything that has mass and occupies space, and states of matter on our planet include solid, liquid, and gas. Solid is defined as some substance that has definite shape and volume. Liquid have definite volume but changeable shape, and gas has both changeable shape and volume. If you take physics, they're gonna tell you there is a fourth state of matter, that is plasma, but it's not relative to our Earth as a planet. So solid, liquid, and gas. So if you have question, what state of matter is water, that would be liquid, all right? What state of matter is ice, that would be solid. Now, energy is capacity to do work or put matter to motion. And two major types of energy is kinetic. It's an energy of action, and potential or stored energy. Now, there is different type of kinetic energy. There is different types of potential energy. One of them that is important for us is chemical energy. So chemical energy that's stored inside those chemical bonds of organic molecules, that's example of potential energy. Now, energy cannot be created or destroyed. We always have the same amount of energy in universe. What we can do, we can convert one form of energy into another form of energy. And this conversion is inefficient because some energy is lost as heat. So when you have your discussions, or when you, for example, have question on your exam, if you say that, you know, mitochondria creates energy, that would be not correct, right? Because if energy cannot be created, we cannot say that mitochondria creates energy or your muscle cells create energy. What they do, they actually synthesize ATP. So they can create ATP because ATP is organic molecule used as energy source, but they cannot create energy. So the whole universe is made of matter and energy. And we just gave definition of matter. Anything that has space and that has mass and occupies space, and energy is ability to do work. Now, matter is composed of elements. Elements cannot be broken down by original chemical means. That what does it mean? Is if you have, for example, gold, right? So that's element of gold. There is no any chemical reaction or any physical processes that you can apply to make it, for example, iron, right? Or mercury. So you have your gold, you will end up with gold. So elements are found in the table of elements. Each element has unique physical properties and chemical properties. Physical properties are measurable, and chemical properties is how atoms interact with one another. Now, elements are made from atoms. So if I have carbon, well, carbon is an element, right? It's found in a periodic table. So carbon is element, but carbon as element is made of carbon atoms. So atoms are unique building blocks of each element, and they have atomic symbol. It's 1 or 2 letter chemical shorthand for each element, and then it has a number on the top that is atomic number, and it has also atomic mass. So we are not going into details about atoms and how they made from these proton, neutrons, electrons, and so on. I don't think so. Let me check very quickly. Yeah, so we're just gonna leave it for chemistry class. We just briefly mentioned that elements are made of atoms, and each atom has atomic number, atomic mass, and symbol, and you can find all the elements in the periodic table. Now, major elements of human body, it's amazing, right? About 96% of your body mass is made of 4 elements only, and it's oxygen, carbon, hydrogen, and nitrogen, right? I actually remember it by the name. The name is Chon, right? So kind of like CHON, right? So Chon is carbon, hydrogen, oxygen, and nitrogen. Those are 4 major elements of human body. About 3% of body mass are lesser elements, including calcium, phosphorus, potassium, sulfur, iodine, iron, magnesium, right, chlorine. So those are elements that make less than 4% of your body mass. And then we have trace elements. They are less than 0.01%. And right, so it's all here like a boron, again, silicon, zinc, right? So those are elements that has we have very small amount of them in our body, right? But they're still essential for normal body functioning. Now, when we have 2 atoms or more than 2, several atoms, they can interact with one another, and this interaction is called chemical bonds. So if we very quickly look at atom of sodium and fluorine here, all atoms, they have protons and neutrons in the core of the atom, right, or nucleus of the atom, and then they have electrons. And electrons are surrounding the nucleus, right, of the atom. So electrons occupy up to 7 electron shells around the nucleus. And octet rule states that except of the first shell, right, so first shell has only 2, can fit only 2 electrons, right? But the rest of electron shells, they can support 8 electrons. So when atom has 8 electrons on its shell, it's a stable atom, right? So it's the atom that does not have this tendency to form any chemical bonds because when we're talking about energy, it's atoms that is stable on its own, right? So each atom wants to achieve this octet rule, so to have 8 electrons in each shell to have this stable energy level, right? That makes sense. The most outer shell is called a valence shell. So if you look at sodium over here, so the first shell has 2 electrons, the second has 8, so this is fulfilled, and the outer shell has only 1 electron, 1 valence electron. So the sodium is not stable in this configuration. Sodium will tend to lose this electron, right? Or to share this electron, or lose this electron. Fluorine, for example, it has 7 valence electrons, right? So it's also has tendency to accept an electron to make it 8, right? So sodium If sodium loses its electron, it would have 8 valence electrons. And if fluorine accepts 1 electron, it would have also 8 valence electrons. And that's what octet rule states, right? So atoms interact in a manner to have 8 electrons in their outermost energy level. So when we have this interaction between electrons, that's what is called chemical bonds. So chemical bonds are interaction between valent electrons. And here's 3 types of chemical bonds. We have more than 3, but those are important in biology: ionic, covalent, and hydrogen. And ionic are mostly formed between metal and non-metal, and covalent between two non-metals, or metalloid and non-metal. Now, ionic bonds form when, let me get a picture for us, right? Instead of just going over slides. Oops. Okay, so ionic bonds are formed when we have 2 atoms with pretty huge difference in their electronegativity. Let's say this chloride atom is more electronegative than sodium. So sodium is losing the electron, right? And chloride accepting 1 electron. So when we have atoms that losing electrons are negatively charged, then atom became positive. And we call it positive ion or cation. When atom accepting 1 extra electron, and again, electrons are negatively charged, so atom became negatively charged, and we call it negative ion or anion. So that's what we have over here. Anion, they have negative charge. Cations, they have positive charge. And when we have positive and negative atoms, they attract to each other. They have tendency to stand together. So that's how they form ionic bonds. And covalent bonds, when we have 2 atoms that sharing their electrons, right? And covalent bonds can be a single covalent bond or double covalent bond, or for example, carbon dioxide, it forms 2 double covalent bonds with 2 atoms of oxygen. And in biochemistry, we represent covalent bonds with lines, right? So you see lines over here. That means that's 2 atoms that share electrons. So now how atoms share their electrons can be equal or unequal. So for example, carbon dioxide, because a carbon and oxygen, their electronegativity is very similar, right? That's why they share electron equally. So electronegativity means the tendency to attract electrons. So if carbon attracts electrons and oxygen attracts electrons with the same force, right? It's like a similar attraction to electrons, that electrons tend to stay, equally shared. And then this molecule has this linear and symmetrical shape, right? And this molecule is called non-polar, because electrons spend their time around oxygen and carbon equally, right? So in overall, the molecule is non-polar. If you look at molecule of water, here we have oxygen and hydrogen, and oxygen is way more electronegative than hydrogen. So oxygen really attracts those electrons way more. So electrons spend most of the time around oxygen. And because electrons are found most of the time close to oxygen, oxygen has partial negative charge, and hydrogens, they have partial positive charge. So this is polar molecule, and polar molecule it has different properties, right? Polar molecule attracts water. This is water, but we have other molecules that are polar. So water and those polar molecules, they will attract each other and they can form these hydrogen bonds. And you can see the molecule is not linear, right? It has this kink, right? It's banded molecule, and oxygen is partially negative and hydrogen is partially positive. So now here kind of like a summary of ionic bond and a polar and non-polar covalent bond, right? So if we have non-polar covalent bond, we have equal sharing of electrons. When we have polar covalent bond, we have unequal sharing of electrons, and ionic bonds are really very polarized covalent bonds, right? So then electrons really spend most of the time around the atoms that is more electronegative, like chloride atom, right? So that's our covalent and ionic bonds. So ionic bonds, atoms, one atom loses electrons and another gains those electrons. They become ions and they attract each other. When we're talking about covalent bonds, atoms share their electrons. Now, hydrogen bonds, very important in biology. Those are the bond between partially charged parts of the molecule, right? What did I say? Okay, so look, if you look over here, we have several water molecules, right? And because water molecules are polar, they have negatively charged end and positively charged end. Now, overall molecule is neutral, but it has partial negative and positive charge, right? And you can imagine that partially this oxygen is attracted to hydrogen, right? So this attraction between oxygen and hydrogen is hydrogen bond, and it's indicated by dotted line, and this is very weak chemical bond, but very important. You will find it in pretty much all organic molecules. You will find it in DNA, you will find it in proteins, right? And so that's attraction between hydrogen and electrically negative atom of another molecule. Now, chemical reaction occurs when chemical bonds are formed, rearranged, or broken. So we mentioned that chemical bonds are attraction between electrons, right? Or interaction between electrons. Chemical bonds are also interaction between different molecules, polar molecules, or even different parts of the same molecule that would be in the protein, different part of protein form these hydrogen bonds with each other. Or in DNA, we have two strands of DNA and we have hydrogen bonds between these two strands. So this attraction between different parts of the same molecule, right? But now when those chemical bonds are formed, rearranged, or broken, that's what we call chemical reaction. And reactions are represented by chemical equations, and chemical equation contains molecular formula for each reactant and amount of reactant and product that should be balanced. Right, so over here we have reactants, hydrogen, and product. So that's a hydrogen atoms forming hydrogen gas, that is a product. Or 4 hydrogen atoms and 1 carbon atom form methane, right, the product. That is the simplest organic molecule, by the way, methane. Okay, now, so what are chemical reactions? Chemical reaction when chemical bonds are broken, formed, or rearranged. And in a chemical reaction, we have reactants and we have products, right? Now, when we look at this chemical reaction, we have exergonic reaction and endergonic reaction. Exer means you exit, right? You go out. Those reactions release energy. And endergonic reaction, those reactions store energy within chemical bonds. Now in biology, so you heard about exergonic and endergonic reaction in chemistry. In biology, example of exergonic reaction are catabolic. So catabolic reaction that when we break large organic molecule into their smaller compounds and energy is released. Now anabolic reaction are endergonic. So it's when we build large organic molecules and store energy. What would be example? Well, example of catabolic reaction would be digestion. So when you eat steak, for example, steak is made of protein. And what happens in your GI tract, you break this protein into smaller molecules that are amino acid. So you break large molecule of protein into smaller molecules. So that's a catabolic reaction. And energy is released, right? So you eat, so you can use this energy, to move your muscles and, to filter your blood, to pump your blood. That's why you need food. That's why you need these catabolic reactions. Now, what would be example of anabolic reaction? When you, when you have those amino acids in your blood, right? Now you can bring those amino acids inside your muscles and make your muscles bigger. So now you're building proteins from those amino acids, right? And you need energy to do it. So it's endergonic reaction or anabolic reaction in biology, right? And you store energy within those large proteins. Okay, so now another thing that we need to remember about endergonic and exergonic reaction is that in endergonic reaction, energy of reactants are lower than the energy of product, right? Because you're storing energy. So energy within this amino acid is less, it's less energy compared with your final product. So in protein, you have lots of energy. You have little energy in amino acids. In exergonic reaction, here you have your proteins and you break it into amino acids, right? So proteins have more energy and your products over here, amino acid, would have less energy. And less energy is released and energy is absorbed. So your whole body works on this endergonic and exergonic reaction, on catabolism and anabolism, because we constantly, our cells break down large molecules and your cells constantly build molecules, right? You break down big molecules to get energy to build your own proteins, your own lipids, right? And your own carbohydrates. Now in biology, it's called metabolism, right? So metabolism, when you take your catabolism and add with anabolism, right, together, you will have metabolism. So metabolism are all chemical reactions that take place in the organism, and catabolism is used to release energy from reactants, and anabolism is used to store energy inside a product. And it's all interconnected through very important molecule ATP. So through catabolism, we release energy to make ATP. Now this ATP can be used to build biopolymers, right? And this is for growth, right? So they connected through this ATP-ADP cycle. Now, example of anabolic reaction would be dehydration synthesis. Synthesis means you're making something bigger, right? Dehydration means water molecule is removed through during this process. So when your cells, your liver cells, for example, they connect glucose molecules together, making glycogen. This is how your body stores carbs as energy source, right? This is dehydration synthesis and anabolic reaction. Also your liver or your adipocytes, your fat cells, they can join or form chemical bonds between glycerol and 3 fatty acid, making triglycerides that you also store in your fat. So again, anabolic reaction. Or your cells combine amino acid in a chain, right? In a primary structure of protein, making polypeptides. So that's also dehydration synthesis anabolic reaction. Or we can put nucleotides together, your cells, making nucleic acid. So that's anabolic reaction. Or catabolic would be hydrolysis. Hydrolysis, lysis means you split something apart, and hydra, water, so you split something using water. So water is consumed and large molecules are split into their smaller parts. That is reverse reaction of anabolism, right? So now glycogen into glucose, triglycerides into fatty acid and glycerol, right? Polypeptides into amino acid, nucleic acid into nucleotides. Now, another example of catabolic reaction is cellular respiration. So when your cells, they break down a glucose molecule in the presence of oxygen, and they use released energy to synthesize ATP. So it's another example of catabolic reaction. Now, rate of chemical reaction depends on temperature, right? Higher the temperature, faster the rate, particle size, smaller the particles, faster the rate of reaction, concentration of reactants. You increase concentration, you increase rate of reaction. Now, in biological organism, right, inside us, inside our cells, reaction cannot happen without enzymes. So enzymes are biological catalysts, and they increase rate of reaction without being chemically changed. So enzymes are biological catalysts. Enzymes are very specific, and for pretty much every single reaction inside your cells, you have a specific enzyme to do it. Now, compounds, very quickly. So compounds can be organic and inorganic. So inorganic example here, water, salt, many acid and bases, inorganic, carbohydrates, fats, proteins, nucleic acid. Now, what is the difference between organic and inorganic compound? Organic compounds are made, they always have carbon and hydrogen, right? Those are two atoms that always present in organic compounds. And then we can have a phosphorus, we can have sulfur, right? We can have nitrogen. But to be an organic compound, this compound must have carbon and hydrogen, and carbon need to be reduced, right? Inorganic compound can, it says do not contain carbon. That's not really correct, because carbon dioxide, for example, considered to be inorganic. It does have carbon, but it doesn't have hydrogen. And carbon is actually oxidized. It's not reduced in carbon dioxide. So just to make it kind of simple, just remember that organic compounds have carbon and hydrogen. Okay, now water is inorganic compound. Water is about 60–80% of volume of living cell, and it's most important inorganic compound in living organism because of unique properties of water that we're not gonna cover over here, because that's really a lot to talk about properties. You can read about it in your chapter 2. Now salts also inorganic compounds. They are ionic compounds that dissociate in water, and they contain cations other than hydrogen and OH minus, right? So everything that dissociate in water and has some cations rather than this H plus and OH minus, that would be your salt. Right, so for example, this is the acid over here, because acid also dissociate, but it dissociate into cation and anion, and cation is hydrogen. Base, it has a OH group, right, minus here. So that cannot be salt. Salt has something different rather than H plus and OH minus. So ions or electrolytes, they conduct electrical currents in a solution and play important role in the body function. We will talk a lot about different ions, especially sodium, potassium, and calcium, because your nervous system, your muscles, your glands, they all require movement, of the sodium, potassium, and calcium for their function. So nervous system cannot function without those ions, right? Muscles cannot function. Nothing can be secreted without calcium. For your glands, endocrine glands to secrete their products, for neurons to secrete neurotransmitters, we need intake of calcium by cells, right? So very important role in biological functions. Now, acids solution contains hydrogen. So when we have increase H plus hydrogen ions, so when we have increase of H plus in the solution, acidity increases. When we have decrease of H plus, then acidity decreases. So to represent how acidic or alkaline solution is, we use pH scale, and it's a negative logarithm of the concentration of hydrogen ions, and neutral solution pH 7, right? And everything that we have over here, acidity goes below 7, that will be acidic. And pH go above 7, right? That it will be basic or alkaline solution. So the lowest pH, more acidic solution is. The highest pH, more alkaline. Right, so over here you can see here's our neutral, and then we go up, that will be base, and we go down, that's going to be acid. Now, pH homeostasis is critical for body functioning, right? And pH is regulated by your kidneys, by your lungs, and by your blood, right? So kidneys can regulate the concentration of ions. You can either remove those, sodium, potassium ions, right, and hydrogen plus hydrogen ions from your blood, or you can retain them. Lungs, you're removing carbon dioxide, right, and buffer in your blood. We will talk about it in the next slide. Those are regulatory mechanisms for normal pH of your body fluids and your cells, right? Because even slight change in pH can be fatal for our cells. Now, what are buffers? Buffers are compounds that resist pH change. And buffers are made from acid and weak base, right? So, so this is the buffer system found in your in the plasma of the blood. It has the carbonic acid, and it has a bicarbonate. So we call it carbonic acid bicarbonate system. Right, so imagine your blood plasma, and in your blood plasma, you have both this weak acid, carbonic acid, and this weak base, that is bicarbonate. Now, what's gonna happen when pH, when your blood plasma became more acidic, what does it mean? It means you have more and more protons, hydrogen ions. So you have more and more and more and more protons, right, in your blood plasma, right? So here you have lots of protons, and that means that pH are actually going down, right? And acidity goes up. Acidity goes up, pH goes down, because you have increased concentration of protons. Now, what's gonna happen? You need to deal with these protons somehow. Well, because you have this buffer in your blood, right? Those protons will be attached to bicarbonate. So bicarbonate gonna accept those hydrogens, and it becomes carbonic acid. So carbonic acid concentration goes up, right? Because your bicarbonate start accepting those protons, right? You have more, it's interesting, you have more acid in your blood, right? You have more carbonic acid in your blood, but actually your blood became more alkaline, because it's not about how much acid you have, it's how much hydrogens you have. Does it make sense? I see somebody has a question for me, so let me go ahead and see the question. Okay, so drinking water that has pH positive or negative, how does that affect the body? Yeah, well, when you drink the water that has, let's say, alkaline water, right? So that make your blood plasma less acidic, right? And if you drink the water that has that is more acidic, that make your blood more acidic, blood plasma. Right? That makes sense. Okay, but because every time when you drink your water, right? You drink your water, you absorb this water in your bloodstream. So it depends what type of water you drink. If it's very acidic water, right? That means it has high concentration of protons. In your small intestine, you're gonna absorb those protons in your bloodstream, and the, of course, acidity of your blood plasma also goes up, right? They do recommend to drink this alkaline water. However, there is not enough research to really, it's fine, it's fine. If you guys need to leave, it's okay, because it's all optional, and please just watch this recording. Right, so so that's the buffers in our blood plasma. Okay, next, let me see. Okay, something. For some reason, hold on a second. Doesn't wanna move anywhere. For a second, I'm stuck over here, guys. So let me just, okay. So, okay, so I was just stuck on this page. So here's our next page. So on this slide, you can see actually how a small change in acidity of your blood plasma, right? Change in blood pH level can affect your body. Well, your normal blood pH is actually 735 to 7.45. So you see, it's a little bit on the alkaline, right? So it's not 7. pH of your blood is not 7. Its pH is a little bit higher, and we go up, we became more basic, right? Remember, go down via acidic, go up via basic. So it's a little bit on the basic side, right? 735, 745. So when we change into 7, it's called acidosis. When it changed to up to 7.8, it's called alkalosis. And below and above this number is death of cells, right? So that's how important to keep pH level balanced, in the body, right? Okay, now organic compounds contains carbon, except carbon dioxide, carbon monoxide that are inorganic, but we can see neither one of them has hydrogen over here. Organic compounds are unique to living system, and in this chapter we will cover carbohydrates, lipids, proteins, and nucleic acids. So organic compounds, many of them are polymers. Polymers means you have repeating blocks that we call monomers, and they bounded together through these covalent bonds. Right? So we have repeating monomers that make a polymer. Now, carbohydrates are polymers, proteins are polymers, nucleic acids, that is your DNA and RNA, are polymers. Lipids are not polymers. Now, polymers are synthesized by dehydration synthesis, and they're broken down by hydrolysis reaction. So here's the example of dehydration synthesis. So you have two monomers, and within your cells, using a very specific enzyme, and enzymes are specific for different reaction, this water molecule are removed and monomers are linked by covalent bonds. That's why it's called dehydration, because water is removed, and synthesis, because monomers are linked. Now, hydrolysis, when we have now this polymer, right, there's two monomers linked together, water is used or consumed to break down these chemical bonds. So that's why we call it hydrolysis. Lysis, we break it down and we use water to do it. So you can see those reactions kind of reverse of each other. And here's example how we have glucose and fructose joined together making sucrose, right? So that would be dehydration synthesis. And then sucrose in your small intestine can be hydrolyzed, broken down into glucose and fructose again, right? And this would be hydrolysis. So sucrose is table sugar, just in case you forgot. So when you put sucrose, your table sugar, in your coffee in the morning, and when you drink it, actually sucrose cannot be absorbed into your blood. And because it cannot be absorbed into your blood, it cannot be used by your cells. Because why would you eat sugar? You eat sugar because you want this simple sugar, monosaccharides, to be used by your cells as energy source. So when you eat lots of sucrose, sucrose cannot move from your guts to your blood. First, it needs to be digested into glucose and fructose, and glucose and fructose now can move to your bloodstream and can be delivered to your cells, and your cells can use it as energy source. Now, carbohydrates include sugar, starches, and some others. Examples of carbohydrates, we will talk about it in a minute. All of them contains carbon, hydrogen, and oxygen, and this is empirical formula CH2O, and you can see carbon and water. That's why it's carbohydrates. And then the number of carbon molecule equals the number of oxygen molecules, and we always have twice hydrogen. So three classes: monosaccharide, disaccharide, and polysaccharides. So function of carbohydrates are mostly as energy source, but some carbohydrates are structural molecules. For example, ribose in RNA or deoxyribose in DNA. So major function is energy or fuel, and also structural molecule, example, pentose, sugar, ribose, and deoxyribose. Now, simple sugar contains 3 to 7 carbon atoms. And here's examples of a simple sugar or monosaccharides: glucose, fructose, and galactose, and 2 pentose sugar. Pentose means 5 carbon atoms, ribose and deoxyribose. Disaccharides are double sugar. So they're too large to pass through a cell membrane. That's why, as I said, you cannot absorb disaccharides. So they need to be digested or hydrolyzed or broken down into monosaccharides. So here's an example of 3 most common: sucrose, that's your table sugar; maltose, found in many drinks, right? And lactose, found in milk. So maltose, we call it grain sugar. So you would find it in beer and any drinks that, that would have something to do with grain to produce them, right? So sucrose, maltose, and lactose. Maltose is two glucose molecules linked together. Sucrose is glucose and fructose, and glucose and galactose together give you lactose. So those molecules have different physical properties and chemical properties. They also differ in their sweetness. Glucose is pretty sweet. Galactose is the least sweet monosaccharide. Galactose, right? So lactose, maltose, lactose is not that sweet. So if you drink milk, you can feel a little bit of sweetness of the lactose, but it way less than sucrose, for example, right? Or maltose even. Now, polysaccharides are polymers of simple sugar. Example would be starch and glycogen. They're not very soluble in water, and we have molecules of glucose joined together, like in the glycogen, for example. And glycogen can also form different branches. And if when you start doing your lab, you will learn more about these organic compounds. This is a nice labs, the labs that we have that explain you different mono, di, and polysaccharides. Now lipids. Look over here, we can see lipids are also made from carbon, hydrogen, and oxygen, the same as carbohydrates. But the difference is they have way less oxygen. And because they have way less oxygen, they are non-polar molecules. And because they're non-polar, they're insoluble in water. So here's the major types. We have triglycerides, another name is neutral fats, phospholipids, steroids, and eicosanoids or eicosanoids. This one might be not familiar to you. I'll explain it, what it is in a second. Now, triglyceride or neutral fats can be solid or liquid, and they all composed of 3 fatty acids bound to 1 glycerol molecule, and major function of triglyceride is energy, storage, insulation, and protection. So this is what we accumulate in our body, right? When we don't exercise enough and we don't eat healthy, and we are not on a healthy diet, that's our regular fat that is stored inside cells that called adipocytes, right? Okay. Now here you can see glycerol molecule, that is organic alcohol, 3 fatty acids, and you can see that they have like lots of carbon and hydrogen, right? And very little oxygen. Through dehydration synthesis, they form covalent bonds and a molecule of neutral fat. Now fatty acids, so those fatty acids over here, because here we have 3 fatty acids. So they can be saturated or unsaturated. Saturated means they have maximum amount of hydrogen attached to carbon. And it would include animal fat, for example, butter. Unsaturated means they do not have maximum amount of hydrogen. And why wouldn't they have maximum amount of hydrogen? Because they form double bond between carbon. And when we form double bond between carbons, that reduces the number of hydrogen atoms. Those unsaturated fatty acids, they are liquid at room temperature and include plant oils, for example, olive oil. So over here, you can see a saturated fat. Each carbon can form 4 chemical bonds. So each carbon here has these 4 lines. So it's connected with the adjacent carbon and 2 hydrogen, right? So each carbon. So this one connected to oxygen and OH group. So we have maximum amount of hydrogen attached to this molecule. Now, when we have unsaturated fat, because we have double bond between 2 carbons, we actually reduce number of hydrogen. Right, so that's what we call good fat, right? And this is what we call bad fat. But it just, the saturated fat can cause some problems with our arteries, right? Like, it can plug our arteries. And that would, of course, affect your blood pressure and perfusion of blood to the tissue. So it's not very healthy, saturated fat. And by the way, you do not need any saturated fat in your diet, because your body, when it needs it, it can make saturated fat or cholesterol from unsaturated fat. So some studies show that we don't need saturated fat in our diet at all. We do need unsaturated fat. Now, trans fat, what is trans fat? It's actually unsaturated, so you can see there is two bonds between carbon, right? But hydrogen is on the opposite side of this double bond. And for some reason, this makes it very dangerous. That elevates your bad cholesterol. Trans fat is formed when we fry stuff or when we try to make solid lipid from unsaturated. Like if you take, for example, your margarine. Margarine, it's an animal fat that was saturated with hydrogen, and it can create trans fat. Okay, now another example of lipids are phospholipids. They are modified triglyceride. So we also have glycerol, but instead of 3 fatty acids, we have 2 fatty acids and a phosphate head or phosphate group that contains phosphorus. So we call it head and a tail, right? So why is it important? Because they make all living cell membrane. So cell membrane are made from mostly phospholipids, and it creates this phospholipid bilayer. Now, this molecule has a head that is polar. Everything that is polar has partial charge, is hydrophilic, so it's attracted to water. And it has those tails that are hydrophobic, so they're non-polar, so they repel water. And because of these double properties of this molecule, it forms these bilayers. Hydrophobic tails, they attracted to each other, and hydrophilic heads attracted to water that is outside the cell, extracellular fluid, and inside the cell, intracellular fluid. So here's the typical structure of phospholipid molecule. So you can see glycerol, we have 2 fatty acids, one is saturated, one is unsaturated, and this is a phosphorus containing group of polar head. And you can see it has a charge. Here's a little plus over here, right? And little minus, right? So that's a polar molecule. And so it's attracted to water, hydrophilic. Now, steroids. Steroids are also lipids. And steroids, they have completely different structure compared to the triglycerides. But all of them has this 4-ring structure, and they interlock. And then it has some side branches here. So example of steroids would be cholesterol, vitamin D, steroid hormones, bile salts. Steroid hormones would include cholesterol, I'm sorry, estrogen, progesterone, testosterone, right? So that's an example of steroid hormones. Now, eicosanoids, a very diverse group, but it's also, they also lipids, right? So we can see that they has carbon, hydrogen, and very little oxygen that makes them non-polar. Very important example of eicosanoids are prostaglandins. Prostaglandins are named by location where they were found. They were first found in a prostate gland. It's a male gland that surrounding the urethra, right, just inferior to urinary bladder. So inside those glands, they found those molecules or compounds first, and they named them prostaglandins. Now, why are they important? They're important because when your cells are injured, right? So when you cut yourself, when you have bacterial infection, when you have viral infection, anything that damaging your cells has a special enzymes that can transform phospholipids, right? Phospholipids, that cell membrane is made from phospholipids, into prostaglandins. So the first, they transform it into AA acid, arachidonic acid, or AA acid. And then this AA acid is converted into prostaglandins. And those prostaglandins, they are inflammatory chemicals. They cause inflammation. And inflammation is redness, worms, swelling, pain, right? So that's what prostaglandins are. We have several inflammatory molecules, and one of them is prostaglandins. Now, aspirin and Advil, those anti-inflammatory drugs, what they do, they block this conversion of phospholipids into prostaglandins, and it's reduce inflammation. Okay, so other lipids in the body will include fat-soluble vitamins, ADEK, A, E, D, and K, and lipoproteins. Lipoproteins are molecules that transport fat in the blood. So here, calciferol or vitamin D is a steroid, right? And it's made from cholesterol in your skin. So this also lipid, requires sunlight for synthesis, secreted into bloodstream. And a calciferol or vitamin D, what it does, it's increase absorption of calcium in the small intestine. And it's added to milk in US to promote calcium absorption. So if, for example, if you have a patient that need calcium, right, if they have decrease of calcium in their bones, you're gonna prescribe them calcium, but always with the calcium, you will prescribe vitamin D as well, because we can eat as many calcium as we want, but if we have vitamin D deficiency, we're not gonna absorb this calcium into our blood. If we cannot absorb it into blood, we cannot deliver it to our bones, and we cannot deposit it in our bone matrix, right? So we need both calcium and vitamin D. Your own skin synthesizes precursor of vitamin D when you are in the sun. That's why it's require light for synthesis. So, but this is for your skin to synthesize a precursor, and then your liver will convert it into a functional vitamin D. Another group, proteins. Proteins are polymers of amino acid. 20 different amino acids form proteins of our body. They joined by peptide bonds. They contains carbon, hydrogen, oxygen, nitrogen, sometimes sulfur. And in all amino acid, we have the same, the part that is the same, right? So this part over here that has alpha carbon, it has amino group and carboxyl group or acidic group. And what is and this and hydrogen atom. So this part is the same. What is different is this R group. And we have 20 different R groups, and we have 20 different amino acids. So when amino acids are linked together, it's very complicated process, protein synthesis. That's, I mean, in general bio, we go over all the steps. But when amino acids are linked together through the peptide bonds, right, it's a hydrolysis, water is released, we form dipeptide. Then we have oligopeptides, oligosaccharides, oligopeptides means less than 100. And then polypeptides, more than 100. So this is the primary structure of protein. So primary structure is the sequence of amino acid in a polypeptide chain. Now, the primary structure then is modified by your cell, so it's assumed this specific shape through the hydrogen bonds. So you see those dots, and it can be either alpha helix or beta pleated sheet. So this is the secondary structure of proteins, right? So it's either a spiral or a sheet. And then tertiary structure, or number 3, is the combination of helices and beta-pleated sheets. So you have this shown here in purple, the beta-pleated sheet, and shown here in yellow, helices. So the combination of 2 of them make a tertiary structure. So this molecule now is a polypeptide. So that's one polypeptide. Now, some proteins, they are made from several polypeptides. So here you see 1 and 2 polypeptides together, and then proteins have quaternary structure. So all proteins have tertiary structure. Not all of them have quaternary structure, but many do. So quaternary structure form a functional protein. Now, proteins can be divided into globular or functional proteins and into fibrous or structural proteins. So globular proteins are compact, spherical, water-soluble, sensitive to environmental changes, and they have specific functional regions or active sites. Example of globular proteins would be antibodies, hormones, chaperones we are not covering over here in this chapter, and enzymes. So just remember antibodies, hormones, and enzymes, right? So those are globular or functional proteins. And fibrous proteins, they are strand-like, they water-insoluble, they stable. An example would be keratin in your hair, in your nails, in your epidermis, elastin collagen. This found, a lot of elastin collagen is found in different type of connective tissue, for example, in your dermis. Contractile fibers such as actin, myosin that you will find inside your muscle cells, right? But this is water-insoluble, stable protein, and this is for structure. So here's an example of collagen, right? That is a fibrous protein, and this is hemoglobin, that is a functional protein or globular protein. Now, protein denature, when proteins are losing their shape, and this happens because of changes in pH or temperature. It can be reversible when normal conditions are restored, or it can be irreversible. And when it's irreversible, it causes extreme damage. So protein is damaged to the degree when it cannot be restored and it cannot function anymore. So the change in temperature is very critical. That's why when your temperature, your body temperature goes above 104, right, Fahrenheit, you need to go to emergency room, right? So why would you need to go to emergency room? Because this change in the temperature start denaturing your proteins, and many enzymes in your body can be damaged. Enzymes are proteins. So when temperature goes up, your body temperature, your enzymes can lose their shape, and as a result, they can lose their function, and it can be irreversible damage. So you cannot restore those enzymes. You need to synthesize them. Your body needs to synthesize new ones, and it takes time, and it can actually cause coma and death, right? So that's protein denature because of temperature increase. And also changes in pH can do same damage. That's why acetosis and alkalosis are so dangerous, right? Because they damage the protein shape. So enzymes are proteins, they're biological catalysts, and what they do, they lower the activation energy and increase the speed of reaction. Millions of reactions happen per minute because of enzymes. So here we have some characteristics of enzymes. They often named for their reactions they catalyze and usually ends in ase, like hydrolase, oxidase. Some functional enzymes are made from protein and cofactor or upper enzyme, and cofactor is either some iron or maybe some vitamin. So over here, we can see shown in purple enzyme, and it has active sites and substrate. Anything that binds to enzyme is called substrate. So substrate bind to active sites. Now enzyme substrate complex is formed, and it's kind of changing the shape, and it's put either the stress on the chemical bonds, or it brings those substrate together to help formation of the chemical bonds, right? But enzyme substrate complex changes the shape, and this catalyzes chemical reaction. In this situation, we have dehydration synthesis, but it can be hydrolysis as well. Now, enzymes inhibitors, very quickly, they're not proteins, but they very critical because many of medications that we use are enzymes inhibitors. So what they do, they prevent substrate from attaching to enzyme. So over here, we have enzyme shown in green, and we have active site, so substrate can bind over here and reaction will happen, right? What if we have another molecule or compound that can bind to this active site, right? Then substrate cannot bind anymore, so we inhibit the reaction. And this is called competitive inhibition, when substrate and enzyme inhibitor compete for the same active site. Or it can be allosteric inhibition, when we have inhibitor binds to any other site of enzyme, but it causes conformational change of the active site, and substrate still cannot bind. Right, so example would be cyanide. Cyanide is very dangerous poison, and it's inhibit enzyme that is important in the cellular respiration, so your cells stop making ATP. And people with cyanide poison can die within a couple minutes. Or acetylcholinesterase inhibitor that inhibit enzymes that cause muscle paralysis. So those enzyme inhibitor, and this is competitive and non-competitive inhibition. As I said, why would we mention it? Because enzymes inhibitors are very important in medicines. For example, antibiotics that saved millions of lives, they are inhibitors of bacterial enzymes. So bacterial cells, they have a special enzymes that allow them to synthesize a molecule called peptidoglycan. So, just without being too complicated, bacterial cell, if it cannot synthesize peptidoglycan, it will die, right? So what antibiotics do, they inhibit that enzyme that is necessary for synthesis of peptidoglycan. So bacteria have non-functioning enzymes, they cannot synthesize specific molecules, so bacteria die. So that's how antibiotic works. Aspirin and Advil, we already mentioned it. It's inhibit enzymes that convert phospholipids into prostaglandins and decrease inflammation, because prostaglandins are inflammatory chemicals. There is another enzyme inhibitor, very popular. It's called Lipitor. And if you ever have patient with high blood pressure, I can guarantee that about maybe 80% or even more patients with high blood pressure will be prescribed Lipitor. And what Lipitor does, it inhibits the enzymes that convert fatty acid into cholesterol. Because very often people with high blood pressure, they have high cholesterol level. And that high cholesterol, that's what causes their blood pressure goes up, because it plugs their arteries. So it's increase the resistance for blood flow. When we increase the resistance, we increase blood pressure. So if patient has high blood pressure, we want to lower this patient bad cholesterol. And how we can do it? Well, how your body make cholesterol? Well, it takes fatty acid, converts it into cholesterol. What if we just inhibit that enzyme? So that's what lipids are gonna do. Okay, so we, just to remind you, we're talking about proteins, right? We talk about primary structure of proteins, secondary, tertiary and quaternary structure. We talk about globular proteins and fibrous proteins. And we mentioned proteins, the nature. We cover enzymes because enzymes are proteins. All right, and then we briefly talk about enzymes inhibitors, because if we can inhibit enzyme, then we can stop some chemical reactions in our bodies that we don't want those reactions to happen. So that's how we use them in the medicine. Now, another example of proteins would be protein hormones. Now, what is interesting about protein hormones? They must be injected. Steroids hormones are not digested by your digestive system. So steroid hormones can be taken orally. Like if somebody, for example, prescribed the birth control pills. Birth control pills, they have the steroid hormones, estrogen, progesterone. So you take them orally. But if you have a diabetic patient, even if it's a small child, you cannot give them any pills, because when you give them insulin, right, insulin is protein hormone. It needs to be injected. Otherwise, it will be like any steak that you eat, you will digest it. Growth hormone is another example of protein hormone. Antibodies are proteins, right? Those are globular functional proteins. They synthesized by special white blood cells that are called B lymphocytes. And we will talk about antibodies in the immune system. Now, the last group, I'm trying to do it quickly because we're already over 1 hour, but the last group of large organic molecules is nucleic acid. Examples would be DNA and RNA. Those are the largest molecules in the body. They contain carbon, oxygen, hydrogen, that is not surprised, right? Nitrogen and phosphorus. Now, building block of nucleic acids are nucleotides. And nucleotides, let's look over here. So here's the example of nucleotide, 2 nucleotides over here. They all made of sugar, right? So this is a sugar, that's a sugar. In DNA, it's a deoxyribose sugar. So it's a pentose, 5 carbon atoms. So sugar, then phosphate group, and nitrogenous base. Right, so those are nucleotides, and the nucleotides are linked together by these covalent bonds, forming 2 strands of DNA or 1 strand of RNA. Now, these strands are held together by hydrogen bonds between the nitrogen bases. Right, so sugar, phosphate, base, that's 1 nucleotide. Now, this nucleotide are linked together, making a chain, right? Or this 1 strand of DNA. And then we have another strand of DNA that made of the chain of nucleotide as well. Now, what holds these 2 strands together are hydrogen bonds between the bases. Now, in DNA and RNA, we have 4 different bases, right? And we abbreviate them with letters A, T, C, and G. The name is adenine, thymine, cytosine, guanine. And there is a special, what we call base pairing rule, or Chargaff's rule. And it states that A always pairs with T and C always pairs with G. So A with T, C with G. Apples in a tree, cars in a garage, and this is what forms this double helix for DNA. Right, so let's see what is in our PowerPoints. So we said over here that 4 bases, DNA is double-stranded, DNA provides instruction for protein synthesis, and DNA replicates before cell division. Now, RNA also has 4 bases, but instead of T, thymine, it has uracil. And RNA is single-stranded molecule, and we have many varieties of RNA, 3 of them important for protein synthesis, such as messenger RNA, transfer RNA, and ribosomal RNA. Right, so that's our RNA. Now, and the last slide over here that we're covering is ATP, right, over here. Now, why all of a sudden we're talking about ATP? Because I'm just reminding you that we were talking about carbohydrates, lipids, proteins, and nucleic acids. So nucleic acids are DNA, RNA. Now, why all of a sudden we're talking about ATP? And this is because ATP is nucleotide as well. It has sugar, ribose, it has nitrogenous base, adenine, and instead of 1 phosphate group, it has 3 phosphate groups. And that's why it's called adenosine triphosphate, 3 phosphate group. Now, what's important about this molecule, that this is the biological source of energy. So inside these bonds between phosphate groups, we have high energy source. That's a high energy source. And when your cells need to do any anabolic reaction, right, if they need energy to synthesize something, they're gonna break those chemical bonds between phosphate groups and use this energy for the cellular work, not only for synthesis, for everything, for muscle movement, for pumping your blood, for, you know, your liver cells gonna use it to synthesize, you know, some organic molecules, your kidney gonna use it to filter your blood, so on. So now when we have this removal of these phosphate groups, if we remove one, we end up with adenosine diphosphate or ADP. When we remove 2, we have adenosine monophosphate or AMP, right? So these 3 groups is ATP, then ADP, and AMP, right? So that's the molecule that is used by your cells for cellular work. And here's our function. So terminal phosphates are enzymatically transferred, and the energy energy is released, and it energizes the molecule. And such prime molecules perform cellular work using the phosphate bond energy. So let's look over here. Here's for 3 examples how your cells can use ATP. Now, first example we will cover in next chapter are those proteins that are called channel. Here's a transport protein. So that's a transport protein. So they're gonna move molecules from extracellular fluid inside the cell. Now, this transport proteins, they use ATP energy. So ATP, 1 phosphate group from ATP is removed and attached to this protein, right? And this is what gives this protein energy to change its shape and move molecules, right? For example, here, right? Again, the concentration gradient. So from ATP, we go to ADP and phosphate group, right? And this phosphate group phosphorylizes those proteins. Here, your muscles are relaxed, and now your muscles need to contract. So it's also gonna happen because of the use of ATP. So phosphate group is released, energy is used for contraction. So here's the chemical work. ATP is used to provide energy for this chemical reaction, right? So this is anabolic reaction. We use energy of ATP to link 2 smaller part of molecule together. Okay, so that's it for this chapter. This was one of the longest chapter, so I apologize, but that's a long lecture. Thank you for being here and thank you for watching. Right, I'm gonna stop recording and you guys can ask your questions if you have any.